Hello, my name is Nick Elder. I'm a fourth-year graduate student in the Developmental and Stem Cell Biology program at UCSF and a member of the V2a Lab. And I'm really excited to share with you today some of my thesis research with the goal of understanding spinal cord development. I'm interested in the spinal cord because it controls several complex functions, such as breathing, motor control, left-right coordination, blood pressure regulation, and bowel and bladder function. Further, the spinal cord performs all of these functions with a limited number of cardinal neuron types that it has to rearrange into different circuits to perform these different tasks. And lastly, the spinal cord can be irreversibly damaged in ways that have lifelong implications, such as paraplegia. In our lab, we use human pluripotent stem cells to generate spinal neurons in a dish. And we do this by providing the stem cells with the same signals that they would normally see during development. And this includes things like Wnt and FGF, retinoic acid, sonic hedgehog, and notch inhibition. Here you can see some examples of these cells both before and after they become neurons. On the left is a colony of human pluripotent stem cells, and on the right are spinal neurons after about 3 weeks of culture using those developmental pathways that I just mentioned. Specifically, I'm interested in a population of neurons called the V2a neurons. These are found throughout the spinal cord and are involved in motor circuits, make direct connections onto the motor neurons. And they're implicated in many of the vital spinal cord functions that I mentioned before. On the right, in this cross-sectional diagram of the spinal cord, you can see where the V2a neurons are born in the lower half of the spinal cord. My research has implications in 3 broad areas that I'll highlight. Those are development, disease modeling, and therapy. During development, spinal neurons express Hox genes, and these act as cellular zip codes within the spinal cord that tell a neuron where it is along the head to tail axis. I'm interested in whether these Hox genes change V2a neuron gene expression or function. A second area that I'm interested in is disease modeling. V2a neurons are involved in motor circuits and could be used to study neurodegenerative diseases such as ALS, where the motor neurons degenerate. And lastly, I'm also interested in the therapeutic potential of V2a neurons, specifically whether these neurons could be transplanted in a way to treat spinal cord injury. If you're interested in learning more, please come check out my poster titled Generating Axially Diverse Populations of Spinal Interneurons for Epigenetic and Functional Characterization. And with that, I'd like to thank my lab, the V2a Lab, and the ARCS Foundation for supporting my research. Thank you.